nociception is the reception of a harmful signal, and pain is actually what the brains make out of this.
Wow. Some also students don't even answer like that. So it's, yeah, indeed.
Actually, I was in your reading on 2.1, and I said the same thing.
I'll bet you remember as well. That's okay. So indeed, nociception, the word ception already said, detection of something, and noci means harmful, so it's any stimulus that can threaten your body, that can make some damage to your body. Of course, that you want to prevent, but let's say if you have a stimulus that can harm your body in any way, you do not necessarily need to mind it. Some people even like it. So it really depends on how you define things, but that is very essential. That this is only, your body is telling you something like, yeah, this will damage you. And the fact that you don't like it, and that's what we probably call a pain experience, that makes you prevent that stimulus from happening, or at least you are not going near that stimulus too often. You are learning from it, also next time, that you should not threaten your body like that. So there is an essential difference. This is the mere detection of a threatening stimulus, and pain is actually, let's say, not only the detection of such a stimulus, but especially the emotional coloring that you get after that. The fact that you dislike it. Anyways, just to go back a little bit, to give you a little historical perspective, although it's also not my main thing, so I have probably not more information about it than the few lines that I will have with these slides. So this is something in the 16th century that you will find in many in textbooks or internet sites when you read about the literature, history, and pain. And this is of course quite a while ago, but at that stage, and that is actually quite interesting, I think, that you see that whatever the main concept of human knowledge at that time was, everything deals around that. And at that stage, everything was, as they call, cardiocentric, so the heart was the center of everything, the center of reason, center of touch, of emotion, anything. So that means that if you had a snake bite, so that drawing, I don't know who made this drawing, but made a nice snake there, at least it draws better than me. I can see the snake, you can also see the fiber. You see that everything it touches something and connects to the heart. So also pain, besides all the other processes, are all going to the heart, and the heart is your conscience or whatever. Of course, they know that we have a brain. But the brain at that time was just regarded as a cooling device, and that you don't get overheated. Okay, then a little while later, they changed the concept, because at that time mechanistics were more popular, becoming more popular. And mechanistics means that they of course start developing machines to help you and not have to suffer so much doing all the efforts. But they also knew that machines, you put something in, and you get a very predictable outcome. Very simple way of thinking. So that's also how they thought that the body works. You just have, like in machines, often works by hydraulics. You have some fluids in there. If you press on fluid from one side, you will see that the fluid comes out the other way. So it's the same thing. We have fluids in our body, and if somehow there is a stimulus at one end, it will somehow pressure the fluid. Fluid, because of the mechanical laws, will just go through the brain. The brain was now actually appreciated, and because we got some pressure on the brain, that's probably why we had a foundation over there. Descartes, René Descartes is probably the person that you may heard of in that context. So, of course, that was actually, then it went quite quickly, and they started to understand that the brain was actually more and more important. So now they know that everything comes into the brain. But of course, not everything hurts. And that means that not everything will actually reach the brain. What do you know of any center that selects information before it falls?
Thalamus.
Yeah, the thalamus, indeed. So the thalamus is actually like a huge filter station, which is just below the cortex and the encephalon. And any sensory information will actually come there, and there it will be decided what goes through the cerebral cortex and what does not. So what gains our consciousness and what does not. Okay. But let's say the nociceptive system is, of course, a protective system because it prevents us from harm. But then the question is how it works. Yeah, that's what I just said. First of all, the stimulus, if you really don't want to be exposed to it, it should be very unpleasant. And if you think about something being unpleasant, what brain structure do you think about? Yeah. Yeah, amygdala for sure. Yes, indeed. So you can mention any component of the limbic system actually, doesn't matter amygdala, or you come up with hypothalamus, or maybe the cingulate cortex or whatever you come up with. But let's say that whole circuit of Papez is involved in emotions. The limbic system is the system that makes us have emotions. Negative emotions or positive emotions, doesn't matter. In this case it's unpleasant, so but still in the same limbic system that is activated. So that means if we have a stimulus that is threatening and that makes you feel pain because it's not good, then it means that suppose that it's in the finger, the painful, the harmful stimulus, that somehow there should be a connection between this peripheral side and the limbic system, and it exists. Have you ever seen, for sure some of you, but I don't know, maybe not all, what is the anatomy of the nociceptive system? So it starts with a nerve fiber in the skin, from something goes to the spinal cord, and to the brain. Let's say, where would the classical ending of the pathway be? What would be the final station for him? Many thanks. Yeah, you could say perhaps motor neurons to withdraw from the stimulus, but I'd say the fact that you have this experience, it's not a motor, it's more a sensory experience. So the brain is telling you something. Where in the brain would you find that? That I would link to the limbic system. Yeah. Let's say usually, I don't know what the rest of the next slide. Yeah. This is the limbic system, by the way. That's just to show you in a picture. So you have all kinds of structures which are connected. It's like a circular system. Makes the emotions go on. Yeah, well, actually you're quite close there. It's indeed the prefrontal cortex, but especially the gut sulcus just before the central, the gyrus before central sulcus. And there you have this kind of guy. You saw it before. The homunculus. And that's what I tried to tell you. That, I will come back to that. Here we do this first because otherwise it doesn't make sense. So you have the pathway, blah blah blah, goes to the, and it usually ends up in the cortex, and there you have a homunculus usually. So that means that in this cortex, primary sensory cortex, it's, let's say, primary sensory cortex because it's the first part of the cortex where a sensation arrives. And there you have a homunculus usually because any cell in the cortex here is associated with a particular body region. We call that a somatotopic organization. And actually it's very simple. You often see that the shape, we'll go back to the shape, it's quite unbalanced. It's not the reality, I hope. But you can see that actually the parts that are really big are the parts that are very sensitive, especially the lips, the tongue, and also the hands. So you have a lot of, let's say, sensory discrimination in there. If you would, let's say, if you would touch your hands on one particular point and go just a little bit away from it, and you would have something very precise to make this touch, and you would be able to discriminate that one would be higher and one would be lower. Whereas if you do the same thing on the arm, your discrimination is far less. It's simply because you have more neurons dealing with the hand compared to the neurons dealing with the arm. So it tells you about sensory discrimination. So that's what we call somatotopy. Somato from body, soma, and topy from position. So the position of the body on your cortex. And the reason why I mention it is that in many of those textbooks you usually see the pain pathway like that. And actually, this is what I would say, but that's you don't need to remember it because it's my interpretation. This is not really pain, a pain pathway. It's more a nociceptive pathway because the cortex doesn't tell me whether I like it or not. I have a limbic system for that. The cortex only tells me whether or not the stimulus is intense or less intense, let's say a degree of intensity, whether I like it or not, just whether it's strong or less strong, and where it's located.
Would you now say it's nociceptive, but just saying it's a harmful feeling to the body? But I think it's also used for just every touch to your body.
Yeah.
If you just touch it normally, you also go to it's the same pathway to the cortex. So it's not only the nociceptive.
No, you're totally right. Yeah. Here I am focusing on the, let's say, the nociceptive pathway, but you will see if you would look at a pathway where tactile information comes in, it moves via different primary afferent fibers. So let's say the first afferents that take a signal towards the central nervous system have a different shape. They also have, for example, specialized end organs here, which are specialized to detect mechanical pressure or stretch and stuff when they get activated. While a nociceptor does not have any special organ, it just has certain proteins that can detect, let's say, cues of damage or something like this. We can talk a little bit more about that later. So let's say the detection neuron is different between touch and nociception. Then when you go to the spinal cord, you will see that the pain fiber, or I would say a nociceptive fiber for nociception, that it actually makes a synapse, then crosses, and then goes towards the brain. Whereas a tactile fiber would not at all make a synapse here, and it would actually immediately go into the dorsal columns, as we call it here, these white matter tracts in the dorsal spinal cord. And it would only make a synapse in the brainstem. And from the brainstem onwards, it would actually also go to the thalamus and to the cortex. So you're totally right that although the beginning of a touch pathway is very different from a nociceptive pathway, that the end organ or the end area is exactly the same one. And that was, let's say, when people start understanding that they also said, is there actually some part of the brain that is specific for pain? So that maybe if we have a person who is really in a lot of pain, that we can, let's say, give him selective treatments on a certain brain area so he can still feel touch, but not the pain. And that is very difficult because there is a huge overlap. There is actually still, although we are very, let's see, you would maybe expect advanced in time in pain research or whatever science, that we would now finally be able to understand with all the imaging studies where exactly is the pain and where is the touch. But there are still people who are saying that there is what they call a pain matrix. That means a network of neurons which are only involved in nociception. Whereas other people say, no, we don't have a pain matrix. It's simply, let's say, the level of activity in our somatosensory cortex. It's another way of looking at it. And there is still no answer, by the way. There are groups that say, no, we have a pain matrix, and other groups say, no, it's all the same structures, and just the kind of activity in those structures, which is different between touch or pain. So it's quite difficult. But you're right, the end organ is the same. But why I mention is that the cortex, do you remember for the cortex you have that homunculus? Did I put it there? Yeah, okay, like that. So here you have that homunculus. So you see that the neurons from here until there, it's quite a large area of the cortex itself. They all deal with sensations of the lips. And when you see that, for example, let's say an arm area, which is approximately the same size as the lip, whereas of course the arm is much bigger than the lips, that tells you something about the discrimination ability of the lips compared to the arm. But important to realize is that the cortex is only telling you something about the intensity of the stimulus and about the location. That you know, I feel a pain on my lip or I feel pain on my arm, but I know where it is. It's only about localization. It doesn't say anything about whether or not you like it or dislike. So for that, and that is not in every textbook, it is in this picture of course, but not in every, is that you also have a connection to emotional centers, to limbic areas. It goes a bit different. And if you really look well, then you will see that this picture, but it's because it's very difficult to find appropriate picture, this picture is actually not even suitable for this story. because you will see that I make a connection with the emotional centers, but actually you see that it's a starting point, and you have arrows going down. But that is not important. What's important is that you will have, if you have a nociceptive system, you will get the signal to the spinal cord via first, or what we call a primary afferent, taking the signal to the spinal cord, and then from the spinal cord, you go to the thalamus, you end up with one projection to the cortex to know that where the stimulus is, and the second connection from the thalamus to the same limbic areas, like insular cortex, like amygdala. I think the amygdala, by the way, is the most important one, so it's good that you mentioned it first. But these tell you something about that you really should prevent that stimulus. Okay. Now something that deals with, what is your name?
Oh.
I try to learn it. So Aaron mentioned, well, the touch system ends here, the nociceptive system ends here as well. There is, although that is totally true, there is several conditions that actually, I mean, it's very bad for the people having the condition, but actually it teaches us a lot about the functionality of the system that can feel normal touch. So they have sensation, but if they touch something that is super damaging, for example, say water of 100 degrees, put their hand in, they can tell you, that's painful. So, and that is simply the reason is that there is something wrong within the nociceptive system that tells them something about, let's say, the affective components, the pathways that go to the limbic areas. There's something wrong there. You also have people who are not even able to touch, to have painful sensation. There is for example, there is genetic, and I think that this is the classical example. Probably I mentioned it in another course also. But there is a condition called CIPA, which means chronic or congenital insensitivity to pain. And those don't learn that, except if you really like it. But CIPA is a condition in which there is a defect, a genetic defect in a growth factor, and the growth that deficit, that deletion in that growth factor, has as a result that these nociceptors, these first nociceptive neurons here, they cannot be valid because they need that growth factor to actually grow like that. So these persons simply miss the first part of the pain pathway, so they really don't feel pain.
This is the DRG.
Yeah, good point. The DRG is abbreviation for dorsal root ganglion, and actually makes sense because its position, it's a ganglion, it's let's say an enlargement within the dorsal root. So normally you have a dorsal root connecting to the spinal cord, and you have a ventral root. And the dorsal root, so the one let's say more on the back, the dorsal root always contains only sensory fibers, so taking signals towards the CNS. Whereas in the ventral root, there you have the motor fibers, which take signals away from the CNS. And let's say it's very important just for anatomical purposes to discriminate the roots, but the roots, right after leaving the spinal cord, actually merge. So within the peripheral nerve itself, you will have them intermingled again, the sensory and the motor fibers. But in the beginning, when they leave the spinal cord, they're separated. The ventral roots for motor fibers and dorsal roots for sensory fibers or pain fibers. So in these cases, you always talk about dorsal root ganglion. You also see that there is here that little thing in there, that the dash with the button on it. What do you think that is? That is indeed the cell body. So the cell body actually has one branch, actually one process, with a peripheral branch and a central branch. This is one cell actually that we're looking at here. And if you talk about the ventral root, then you have motor neurons. Where would their cell body be of motor neurons? Suppose that you would have here also drawn a ventral root. Is there also a thickening in the ventral root?
They
No,
go straight,
it doesn't exist.
straight through.
Yeah, the signal goes from the CNS towards the PNS, so like that. But where is the cell body of the cell?
Like this is
So
great.
from the spinal cord.
Yeah. It is actually just in the ventral horn of the spinal cord. So you have a cell right here, and that cell projects through the ventral root towards muscle. Of course, there is also motor neurons in the cerebral cortex, and they project down towards the ventral horn, where they make a synapse with these. And actually, if you talk about that, I mean, this is totally not a topic, it's just because of the question comes. But the ones, the motor neurons in the cortex, which are projecting down towards the spinal cord, they are called the upper motor neurons. And the ones in the spinal ventral horn are called the lower motor neurons. Or alpha motor neurons, but forget that. So anyways, that's about the system. So in the, let's say, the nociceptive pathway or the pain pathway, we have the detection of a harmful stimulus in the periphery. And periphery can be anything. It can be internal organs, can also be skin, it can be anything. A signal is communicated to the CNS and then towards higher brain centers, either ending up in the cortex to localize the stimulus and to know whether it's intense or less intense, and a connection to emotional centers, limbic system, to tell it that it's unpleasant.
Okay.
Oh, sorry, yes.
Got a question. Now you show two different pathways. They split up, one for the localization of the cortex, and one for the emotions.
Yeah.
And let's say that you've got a stimulus that's coming in, going to the thalamus, but the thalamus is, let's say it's below threshold, so it doesn't project it into the cortex. But it's higher, but it's still reaching the emotional center. Would that be possible? That you like receive a sensation or something that comes into your emotional center, but that does not reach your brain, so you don't know where it comes from, but it can change your emotion.
I don't know, perhaps. That would be philosophical almost. I don't know what was that, but I can imagine that if there is a stimulus around, and maybe you're not consciously aware of the stimulus because maybe you're distracted somehow, that maybe the, let's say, the pathway towards the cortex is blocked, and you're not aware. But actually you do feel bad, perhaps, because there is some signal. But I think that it's so difficult to investigate this that we don't know a lot about that. But why not? Theoretically, it's possible, simply because, let's say, actually, I need to make sure that you all see that, because that is actually confusing. I'll delete that from the next time. This here, this whole line, forget it, it's not there. Because this is actually just a line that tells you if you have a lot of emotions, then the emotional centers send down signals to inhibit pain. So the only thing that I've told you about so far is just a system that takes signals towards the brain. So the one to the cortex is shown here. The one to the emotional centers is actually not shown at all, but it would be if it would be shown like that, a line towards the emotional centers. From the thalamus, yes. And that's why I actually start saying this now, because of your question. You have different thalamic nuclei. Every nucleus serving a specific purpose. And you have a certain nucleus within the thalamus, which is called the VPL. And usually these nuclei are all called for their position. Ventral, posterior, or lateral nucleus. So it's a little bit ventral, a little bit lateral, and a little bit to the back. And then we call it the VPL. And that one projects to the cortex. But you also have a certain nucleus called the interlaminar cortex, because it's just between some lamina and the thalamus. And that one projects to the emotional centers. So theoretically, it could be that the thalamus is not homogeneously inhibited. And you get, let's say, some active signals towards maybe emotional centers, but not to cortex. But that is theoretical. Other questions so far? I need to make sure that I keep track of the time. All right, let's continue with that. Oh no, wait, I need to. So, oh yeah, sure.
Just go a little bit back at the top of what Joosen said.
Yeah.
There is a Chinese mortal method where they put a drop on your head, and at the beginning it is not intense enough to feel pain,
Yeah.
but after an hour it's getting pain.
Okay.
Isn't that not something like how something of accumulating the intensity in the cortex or something like that?
Possibly. I don't know exactly this technique, but I mean, but this is very theoretical, and I don't think it will apply to your example. There is something that they call wind-up. So if you have a stimulus which is below threshold, if there is a second stimulus shortly after the first, before actually the first one totally diminished, you get a build-up and a build-up, and at a certain moment you go above threshold, and then it starts hurting. But this is all in time frames of milliseconds. And you probably don't, you really mean probably that really in the beginning, maybe minutes, maybe hours, I don't know. It doesn't, so that should be a different phenomenon. I guess you can get some sensitization that we'll also see later. But I would need to read a little bit also about how it works. What is actually the substance that they put there?
It's just a drop of water on your front head.
Is it constantly or just one drop?
Yeah,
If it constantly drop.
But what is the drop?
Then it becomes
Just
just
water.
water.
Just water.
Yeah.
At the beginning, it's yeah, you just notice it is water, but after a half an hour, something like people get really crazy and say it's just like a stone putting on their head.
Actually, I think it's more
Hitting somebody
rather-
constantly
Yeah, yeah.
on the side of his head.
I never heard that.
More on his
I'll
head.
try.
I'll let you know the video.
You can do that for the next
Yeah,
hour.
yeah, yeah.
Actually, we stopped the whole session. This will be the next session.
Screw the MTT practicals, let's do that.
So, let's say, if you want to research things, because of course they matter, and because of pain is not bad at all, but if you have chronic pain and you have pain without a real clear reason, then it becomes actually quite annoying. You need to find something for it. So you start looking into what explains pain, or let's say more pathological pain, chronic pain. And let's say if you start research, it's very important that everybody who will do the research does the research by the same definitions. Not that we're talking about totally different things, especially if you talk about something that is as subjective as pain, because everybody will experience pain in a different way probably. So for that reason, scientists all over the world, scientists, clinicians, whatever, they form groups. You will see that in the neuroscience field. You also have it specifically for the pain field. And those groups are there to, let's say, make a coherency between the scientific interests of the different labs or hospitals. And do you know the group that makes all these things in neuroscience? Do you know what is their name? The groups that join together to say, we are can maybe say not the NATO, something like that, important big organization. But the big organization has a specific goal, and that is to do pain research. In this case, it's called the IASP. That's why it's there. But for neuroscience, you have different ones. You have the mondial one, so all over the world, which is of course funded in America. That's everything. And that is called the Society for Neuroscience, SFN. If you are interested in neuroscience, you for sure remember that, because you will throw into death with it. You also have the European Society, which is called the FANS. Maybe you never heard about it. But anyways, so the reason why I say it, you have all groups who are joined together, and the one for pain is IASP. It stands for International Association, because it's against society, International Association for the Study of Pain. Another very original name.
Excuse me, is the one that is going to have a congress this August in Milan?
Yeah. So that is, let's say the IASP has their annual meetings. So that's when all the IASP members and non-members, anybody who wants to see what is the latest status of research, they are getting together there, and there will be seminars and poster presentations and workshops, whatever what. It costs a lot.
It's terrific.
Yeah, it's very nice. But I mean, here it's about the IASP. You have also other pain groups. Like you have, let's say, the IASP, because it's pain. Pain is, of course, super broad. You have so many different types of pain. You have visceral pain, and like chronic belly pain or something. Or you have nerve injury pain and neuropathic pain. Or you have oncological pain if you have a cancer and you have pain. So it's all different. And you have different societies, and there again, so the IASP also has what they call special interest groups. So these special interest groups, they have their own meetings. And it's quite complex, but the IASP is the mondial one. And that is indeed in Milan, in August. So, if you look at the website of the IASP, you will see a definition for pain, and it says, well, pain is of course a sensory emotional experience, but it needs to be unpleasant, and it needs to be associated with, and then it's very important, although it's not in green, actual or potential tissue damage. So the stimulus that hurts should, in theory, have the capacity to do damage. So even if it doesn't do damage, if the stimulus would maintain and cause damage, then it should definitely hurt. So that's also the way that we learn from it.
But this is physical pain.
And that is a very good point. That's also almost never raised. No, I don't believe that it is only physical. Because damage is one of those words where I personally think it is like, let's say, if I tell you that tomorrow you will be evaluated by experts, because I sometimes do that with my students, I say, to make them a little bit stressed, you know. It's important that because they need to learn also to perform in front of maybe a decent audience. So I say, there will be a jury of experts, and then all the students. Stressed. But, I mean, what does that mean, the word expert? Maybe I'm an expert when I take my cereal in the morning because I do it all. I'm an expert at it. You know, I never said expert at what. And the same is with the word damage. What is damage? Damage is a very unspecific word. It can mean so many things. And personally, I think that if somebody socially, if my girlfriend wants to say something totally bad about me, probably I will feel more hurt or damaged than if she would hit me. But what I mean to say is damage can of course also be psychological damage.
Or why does it say tissue?
That's specific.
Sorry, I didn't listen. No, then you're right. But there also maybe, that's actually good that you mentioned that as well. I didn't see it. But you will see that, I mean, there's this taxonomies. Taxonomies change all the time. We also have taxonomies for neuropathic pain because it's the most severe type of pain. And every 3 years, they adjust the definition because they say, I actually forgot to include that. It's not specific enough, so you need to change it again. So I agree with you that, let's say, if you talk about pain research, most of the research on pain is on pain after a physical stimulus. And you can see that now in the last 5 years or so, maybe a little bit longer, there's more and more groups which are starting to appreciate also social pain, especially, let's say, pain after rejection or something like this. And they put people in the MRI, and they're looking whether the same brain regions are activated compared to if you really have a physical pain. And you see, oh, it's quite similar. But it's let's say, an area under development. And that's probably why it's not included yet, because actually I thought it could be included, but indeed you're right, with tissue damage. Yeah, so, okay. So anyways, that's about the definition. And we have that. Now, of course, if you have a pain, and here I am talking about physical pain, because it's simply what we focus on all the time. Of course, the stimulus starts in the periphery, because that's probably where the harmful stimulus is. So that means that our nociceptors should be different than the one from touch. Like we said, we have the mechanical bodies or the Chini bodies, I don't know, special organs at these endings which are able to detect mechanical changes in the skin. But we also should have, let's say, a specialized site here at the end, which can detect something that is, let's say, a damage cue. Now, what could you imagine? Let's say, if I say that cell here should be able to detect something damaging, how should I equip that cell at that end? What kind of things does it need to have? Yeah.
Threshold?
Yeah, it needs to have a certain threshold, but that applies to any neuron if you want to activate it. But what, let's say, what should it be able to detect? Say damage. Damage is something.
Temperature.
Good. temperature will be what? If the temperature is too high, the thing should be activated. And
Pressure, mechanical, temperature, and
Indeed,
chemical.
if let's say if there is a mechanical stretch or something which is above a certain threshold and you really get tearing of tissue, that should some mechanical stuff. Indeed, something else.
If you have something toxic or acid.
Exactly, acid's super important. So all these things, and I'm telling you, an acid is of course something totally different than, let's say, a mechanical stimulus. So you should have all different types of detectors there that leads to an activation of that cell. Now, what do you think a cell has to detect acid or to detect temperature, or is it because it's not a specialized organ? It's not like a little organ of fibers that if they're stretched, the fibers will actually extend, and like in a muscle spindle, and you get some electrical current towards the spine. It doesn't work like that. There is no special. It's as we call it, a free nerve ending. So it's just like an axon, which is say just having the membrane. And that's it. So the membrane should contain something. What should the membrane contain to detect things like acid or temperature?
Receptors.
Yeah. Protein receptors. So you have specialized proteins, just molecules, which are within the membrane, as there is proteins in any cell's membrane. But these proteins are specifically adapted to detect such a stimulus. Now, how does that work, you think? How can I have, let's say, suppose that my arm is the nerve ending. How do I have, suppose that I have very ugly zip on there, that is the protein. How can that protein, not how it can be done, but suppose that protein can very specifically bind an acid. Now how would that lead to a current along the fiber to the spinal cord?
Iron.
Exactly. So you will see that almost any of the receptor proteins that we're talking about are actually ion channels. At the moment that they will bind their ligand, or they will simply detect their stimulus, they will open their gate, the channel, the channel pore, and you will get fluxes of ions through the channel. And that leads to depolarizations, and the signal goes up to the spinal cord or wherever it needs to be. So that's important to realize. And I give you one example. So here you see the free nerve ending in the skin. And here you also see, with a little imagination, you see that there are pores in these things. The pores are normally closed. But if a ligand binds or whatever, then it opens. We get sodium and potassium channels fluxing through the channel, which leads to a change in the potential of the membrane. And if that depolarization, if it's strong enough, it is above threshold, we get an action potential. And simply because you only have these kind of channels at the peripheral ending, which are there to, let's say, which are there to actually, let's say, give a depolarization which is above threshold. But once you are above threshold, you need other types of channels which actually conduct the signal along the fiber. And that is not at all ion channels that are activated by these acids or whatever, no. That is ion channels which are activated by voltage differences. Voltage-gated ion channels. And they are a little bit different. You usually see them indicated with a little V below from voltage. So in this case it's usually sodium channels which are voltage operated. Okay, so you can, for example, see, just because you made that remark, acid, it's an acid sensitizer, acid sensor. So this ion channel would be opened if there is an acid around. So just one of the examples. The most famous receptor in pain research, if you talk about the detectors in the skin, is the TRPV1. It's what they call the vanilloid receptor, TRPV1. It looks a little bit complicated name. When I saw it the first time, I thought, oh man, I need to memorize that. But actually, when you hear it 50 million times, you start remembering it. But TRPV1 is a channel which is activated by heat above 43 degrees. So if you have temperatures above 43 degrees, ion channel opens, we get fluxes again, we get a depolarization strong enough to activate the voltage-gated sodium channels, and the signal goes to the spinal cord. Do you have any reasons, because I also indicate these red chili peppers, why are they there?
They got, I don't know what it's called.
Capsule.
That was
Ah, you already know.
silicon for the receptor.
Yeah. So there is, within these peppers, there is a certain molecule called indeed capsaicin. And capsaicin also activates the TRPV1 receptor, just like temperatures above 43 degrees does. And that's why hot chili peppers actually are hot. They give the same sensation as temperature above 43 degrees would give you, because they activate the same proteins, and that activates the same neurons, and ends up in the same part of the brain.
43 degrees?
So it's just, let's say, a happy coincidence if you like hot chili peppers, and unhappy if you don't like them. So it's just one receptor which is activated by different stimuli, either by capsaicin, which is active ingredient in these peppers,